Friends, today we are going nucleic acid part 2. In this lecture, we'll study about the experiments that prove that DNA is a genetic material. Now, first of all, the scientist Friedrich Mischer in 1868, what he did, he isolated a phosphorus containing substance which he called nuclein, which we today know as DNA, from the nuclei of pus cell, which was obtained from the discarded surgical bandages. He experimented and found that this nuclein contained an acidic substance, which we today know as DNA, and a basic substance, that is protein. Now, the same acidic substance he also found in the head of sperm cells. But during that time, he was unaware about the chemical structure of DNA, which was identified from late 90s. Now, the first scientist who proved that DNA is a genetic material or DNA carries the information of heredity is come from Griffith in 1928. What he did is he worked on Streptococcus pneumoniae, which causes pneumonia. Now, he used two strains of Streptococcus pneumoniae, virulent strain and avirulent strain. Virulent strain means which can cause disease. Avirulent strain means which cannot cause disease. Why the virulent strain can cause disease? Because it is encapsulated. That means it is protected inside a capsule. That's why it is virulent in nature. Avirulent is non-capsulated. That means it is not protected inside a capsule. Hence, it is avirulent because once it enter our body digests this strain because it is not capsulated or it is not protected. Virulent strain is protected, hence it can cause disease. So what he did, he used two strain. One is virulent strain and another one is avirulent strain. Now what he did, he infected this virulent strain in a mice. What will be the result? The mice will die. Now, next what he did, he injected the avirulent strain in the mice. The result is mice survived. Now what he did, he injected heat-killed virulent strain. Because it is heat-killed, that means it cannot cause a disease now. Though it is a virulent strain, but now it lost its disease-causing capacity. So, the heat-kill strain, when injected in a mice, the result is the mice survived because now the virulent strain cannot cause disease. Now what he did, he mixed heat-killed virulent strain plus avirulent strain and then he injected in a mice. What should be the result? Heat-killed cannot cause disease, avirulent cannot cause disease, the mice should survive. But the shocking result was the mice died. So now the question is, why the mice died when both the strain now cannot cause disease. So that means something is there in the V strain which is transferred into the A strain, making it pathogenic. So Griffith said the substance that is transferred is DNA, which carries the genetic material and hence the pathogenicity also transferred to the A strain. But that time many doesn't agree with the Griffith conclusion because they are in a doubt that how come we conclude that DNA is only carrying the information because the strain also has the protein. So that protein can also carry the information. So others were not convinced by the Griffith conclusion. Now, the next group of scientists worked in 1944, that is Avery, MacLeod, and McCarthy, and it is the extension of the Griffith experiment. What is the last conclusion of the Griffith experiment? What he used? Heat killed virulent strain plus avirulent strain. When injected in mice, the mice should survive, but it died. Now, what these scientists did, they isolated the DNA from heat killed virulent strain and purified it as much as possible. Then they used this DNA and a avirulent strain injected into the mice, and the result is mice died. So by this experiment, by the result of this experiment, they proved that DNA is the genetic material. But again, many are not convinced by his result as well, because the DNA is purified as much as possible. That means the impurity of protein is still present. So maybe in a very minute quantity, but still it is present. That means it also can be a chance that this minute protein is transferring the message to the A strain. So now how to prove that the message is transferred by DNA only? So now what they did is they used DNA plus protein plus protease. What they did, they add protease and then injected into mice. So that means because of this protease, the protein should have gone degraded. So that means only DNA left. So the mice died. Next, what they did, DNA plus protein plus DNase and then injected inside the mice. So the DNase, what it did, it degraded the DNA. So that means now left with only protein. And the result obtained was the mice survived. So that means by this experiment, they proved that it is only the DNA material that carries the genetic information. It is not the protein that carries the genetic information, because if it would have been the protein, in that case, when DNA was added, protein was active, so the mice should have died then. So this experiment proved that the genetic material is present in DNA and DNA is transferred from virulent strain to avirulent strain, making the avirulent strain a pathogenic mnemonic. The next experiment was performed in 1952 by Hershey and Chase. They worked with bacteriophage T2 that infects E. coli. What they did, they used two separate suspension having E. coli, and they infected that E. coli with bacteriophage. Now, in the first case, they labeled the bacteriophage with 32P. And in the second, they labeled the bacteriophage with 35S. So, two set, one with 32P and one with 35S. What will happen? They will infect this bacteria. And what bacteriophage do to infect? They insert their genetic material inside the bacteria. So in this case, they'll inject their genetic material. The red one is labeled with 32P. The blue bacteriophage is labeled with 35S. That means this labeled radioactive sulfur is present in the coat protein and not in the DNA. Next, what they did, they agitated in a blender so that to separate the empty capsid or the empty head of the bacteriophage, which is known as ghost. Then they performed centrifugation to separate the empty bacteriophage head from the bacterial culture. As because they used radio-labeled phosphorescent sulfur, hence they found that here in this set the sulfur is visible because it is radio-labeled. So here sulfur is visible, whereas in the bacterial set they found that DNA is visible. So because sulfur is visible in the part of bacteriophage that is not entered into the bacteria, that means the protein is not a genetic material, because the head of bacteriophage or the shell of bacteriophage or this capsid is made up of protein. Because they found the radio-labeled protein in this empty ghost and not inside the bacteria, that means the bacteriophage has not inserted the protein in the bacteria. What they inserted? Their DNA. That means DNA is the genetic material and not the protein. Clear? So these experiments prove that DNA carries the hereditary information, or DNA is the genetic material. So now one question from my end for you all, that is why 32Ps get incorporated into DNA and 35S into viral capsid protein. Do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.